All right, I think everything is set up and running. So welcome to the next Tuesday seminar at the University of Warsaw Astronomical Observatory. Today is our last seminar this semester, and we end our series of talks this semester, which were going on the topics were related to solar system, planetary systems, stars, sun, and the stellar origins. So we end with a big bang, not literally, but today we have a special guest who is Tomasz Zajkowski, who is a biologist. I don't remember we've had a biologist here at our seminar so far, but Tomasz is working on the in NASA, and is working on biology and the connection between biology and space and origins of life. And these topics, I think, would be of much interest to many of us. Here is Tomasz. Tomasz, the floor is yours, please.
Hello, everyone. Yes, my name is Tomasz Zajkowski, and I am molecular biologist. I'm also astrobiologist and a president of Polish Astrobiology Society that we started. So I will be talking about the Polish Astrobiology Society at the very end of the talk. I will start my presentation with describing the theories about the origin of life, because this is what my main project is about, but I will also mention some projects in the synthetic biology and the space exploration that I'm also working on at NASA Ames Research Center in California, where I lead several independent projects that are mostly related to the origin of life. And that's why today this subject about this fascinating field of research. And during today's meeting, I will be talking about fundamental questions that we ask about the beginnings of life in the universe and beginnings of life on Earth. And here is a set of questions, like how did life begin? We can divide into sub-questions, like when did life begin? What actually is life? Important question of chicken or egg? What was the first? And where exactly did life begin? So I will start this presentation with Andy Warhol's soup. And this is because it is impossible to talk about the origins of life without mentioning the primordial soup. So in 1920, Oparin and Haldane, they described their theories in which they imagined that life could have started in a body of a liquid water full of organic matter. And in this, as they called it, primordial soup, something called abiogenesis was supposed to occur. But this idea of abiogenesis, so the origin outside of the life, is actually a much older concept and reaches back to Darwin. And Darwin in 1871, in one of his letters to his friends, he introduced the concept of a warm little pond that you might have heard about too. But this kind of ideas had no scientific support until Miller and Urey in 1952. They conducted their first famous experiment in which they simulated conditions on early Earth, where they mixed water, hydrogen, methane, ammonia, so reduced gases, and they passed them through glass tubes, treating them with electric discharge. And somehow they managed to avoid blowing up the whole laboratory. And on the third day of the experiment, there was a black tar on the sides of the glass. And they isolated from this tar organic compounds. And one of them were amino acids, so the building blocks of proteins and a major component of life. These were the beginnings of a field called prebiotic chemistry. Their experiments in the very different conditions and various variants are still carried out until today. Even a recent publication from 2020 was actually showing something about maybe possible catalysis of a glass itself. So it's constantly evolving experiment. And in one of the versions, when laser was used instead of electricity, they were able to produce nucleotides, so the basic building blocks of DNA and RNA. So now we have amino acids and we have nucleotides in the same experiment. But the most important thing about the Miller and Urey experiment is that from this boring non-living matter, we can easily create something interesting that is a basis for life. But yet, it's even the simplest bacterium that we can find is still extremely complex, and it is difficult to imagine that it could arise from a simple experiment like Miller and Urey experiment. There must have been some intermediate steps that did not survive until today, so we don't see them. But because everything on Earth is related to each other, some clues about how the first living organism looked like could have survived, and we know they survived in the organisms that live today. So today I would like to take you on a journey to the beginnings of life. And as I mentioned, we will go through these questions, and we don't have the answer to the first one, how did life begin? But we have a lot of hints as what could have happened, and we know what certainly did not happen. So let's start with the first question, first specific question, when did life begin? And just build this frame for concepts like how we can put borders to where life started and what things must have happened. In a very straightforward way, obviously, life on Earth could not have started before Earth itself was formed. And it was formed about 4,456,000,000 years ago. And just an astronomical blink of an eye after that, after the planet was formed, it collided with another planet about the size of Mars. And this event melted the crust and led to the formation of our moon. When the Earth crust cooled again, there might have already been water on it, but not for long, because hundreds of thousands probably of massive meteorites fell on Earth over the next several 100 million years. And this period was called the Great Bombardment. As a result of collisions, the oceans again evaporated and the Earth crust melted. At that time, the Earth just wasn't a good place to live. At least until the things calmed down a little bit, about 4 billion years ago. And this is the earliest possible time of life to begin on our planet. So fossil and chemical evidence show that microorganisms already existed on Earth about 3.7 billion years ago. And that means that about that time, this boring non-living matter became alive. And this is exactly what we would like to understand today. There are also some other controversial evidence that life on Earth was already present 4.1 billion years ago. In this photo, you can see graphite embedded in a mineral called zircon. And this graphite has a carbon isotope typical for living organisms, but it is not a strong evidence for life. Microfossil are the stronger evidence. And here are some examples on the photo. But also this photo shows you that even if we were able to go back in time and look for first organisms, it might be really problematic to recognize them, which brings us to the next question: what actually is life? So what beginnings of what are we searching and trying to understand? So you should expect that the biologists have a decent definition of life. After all, they study life, right? But as a biologist, I must confess that we actually don't. And Haldane, the person that I mentioned at the beginning of the seminar, one of the originators of the concept of primordial soup, in one of his books, he asked the question, what is life? And almost immediately he admitted that he has no answer to this question either. So what exactly is the trouble? Perhaps the problem in defining life is due to the fact that we formulate the question incorrectly. Perhaps life is not something that a living organism have, but rather something that living organisms do. So instead of asking if something is alive, we should ask how much it is alive. Maybe there is a continuity between individual ecosystem, even a whole biosphere. Have you ever thought about Earth as a living organism? So there is a saying that life is something that we will recognize when we see it. Like this microphage chasing bacterium on the image. But let's step back and try to create our own relatively simple definition of life. So in many schools, there are classes held during which teachers make a list of features that life must have. And yet we can find examples of all these features that the non-living things also can do. And they're definitely not alive. And also, the early life didn't actually have to do all of these things. So fortunately, a cat-loving physicist came to aid biologists, and he looked at what living organisms do from the physical perspective. One of the things that he noticed is that according to the second law of thermodynamics, the universe tends to increase its entropy. The energy and matter tends to dissipate and become simpler and simpler in the whole universe. And yet inside a living cell, everything seems to be highly complex and highly ordered. So Schrödinger defined life as a process of counteracting entropy or maintaining disequilibrium. So in general, Schrödinger, he perceived the living organisms as closed chemical systems that worked to maintain order. But the trouble is that this definition ignores one very important feature of living organisms, that is that organisms evolve. Let's take a little bit closer look at the evolvement issue. So we assume that the first living things must have had some information stored in some way, instruction on how to build its individual components and how to copy itself. These molecules are presumed to be polymers because that's what the current cells use. And polymers are nothing less than just big structures made of smaller components, as we call subunits. So here is an example of a biological polymer in a modern cell. It's an actin filament, part of a cytoskeleton that was recently found even in bacteria. So the actin doesn't store information, but it's just a good example of how subunits build a longer chain. And you can imagine the same thing happening with a DNA or RNA. And a similar phenomenon occurs on a smaller scale, also outside of the cell. And in this example, this is an growing RNA chain. It polymerizes on a surface of clay. The chain is formed from nucleotides from the environment. They are attached in a random order, so resulting chains are different from one another, and they interact differently with the environment. Some of them are maybe more efficient in obtaining other and the components and their building material, and so they exhaust the resource of subunits, limiting the growth of their competition. And still, we're talking about just a simple chemicals on a mineral. And what I just described is already a Darwinian evolution by natural selection. So for life to develop, evolve, and even originate, the competition or natural selection had to exist from the very beginning. In chemistry, this process is called chemical evolution. So we established that life maintains order in disordered environment and is a product of evolution. Life probably began when molecules began to copy themselves and become more and more efficient in making copies through natural selection. This process started in the chemical environment even before the first cells became recognizable. But of course, NASA needs some working definition. If we look for life, we need to know what are we looking at, otherwise, how can we justify our grants? So Gerald Joyce, he created this working definition, that is, life is a self-sustaining chemical system capable of Darwinian evolution. So this is what NASA is looking outside of our planet, for example, on Mars. So we already have a working definition of life. Using it, we can begin to describe what should characterize the organisms that we are looking for, either on early Earth or on other planets or moons. So life should counteract entropy, and to do this, it should create a closed system, for example, be made of cells. It should have some kind of molecule capable of carrying information on how to build its own components, and this information should be able to evolve through natural selection. Let's see how it's done in our own cells. When we look at living organisms, one of the universal properties of all life is that the most of the cellular machinery is made of proteins. So proteins are the functional elements of cells. Our cells, like all known organisms on our planet, when they produce proteins, they must copy genes from DNA to RNA and use RNA as a blueprint or a template for protein production. And this universal direction of this mechanism is called central dogma of molecular biology. And so we come to the question that I hope everyone were waiting to ask the most. What was the first, the DNA or the proteins? So in the central dogma of molecular biology, there is this paradox, the sort of a problem of what came first. DNA needs proteins to replicate itself. Cells use DNA to produce proteins. So what was first? Fortunately, when we consider the problem of the origin of life, we can solve this paradox quite easily. The solution is to get rid of DNA and proteins, and let's just leave everything to RNA. RNA is a molecule related to DNA. It contains the same 4 letters of genetic code, with the difference that the thymine is replaced with chemically similar compound uracil, and instead of 2 strands and helix, RNA is usually just 1 strand. And RNA is unique because in addition to its ability to carry information, it can also fold into complex shapes, very similar to what proteins do. And this shape enables them to perform functions, just like proteins do. For example, one of the functions could be catalysis. They can catalyze chemical reactions, or they can catalyze self-replication. So a popular theory of the origin of life is called the RNA world hypothesis, that is recently starting being dominated by RNA peptide word hypothesis, because people started putting more value on the presence of peptides in prebiotic, let's say, pounds, like warm little pounds. So the word before proteins evolved was called the RNA word. And in the RNA word, the RNA performed both information and catalytical functions. And for this theory to be true, even if the RNA word is no longer here, we should still be able to produce RNA-based enzymes or ribozymes, just synthetically in laboratories. And indeed, in the laboratories, we can produce artificial ribozymes, so RNA enzymes called ribozymes. And these RNA ribozymes, they're able to copy their own sequence. And copying this sequence is not 100% accurate, so it has errors. And that's important because errors are the basis for natural selection. Another strong argument in favor of the RNA world hypothesis is the fact that the ribosome, that is the molecular machine that is used to make proteins, it is a protein that produces other proteins based on DNA. Information in DNA passed to RNA, so actually on RNA. And this protein itself, if we just look at the very core of it, the center of the ribosome is a ribozyme, so an RNA. Catalytical RNA constitutes the oldest central part of the protein that makes other proteins. So another strong argument for the origin of life starting from RNA. In addition, nucleotides, so the components of RNA, are made, so RNA, it's made of parts, of the components of nucleotides. And these nucleotides are also used in metabolism, in many metabolic processes. For example, ATP, that is shown here, stores energy for living cells. So the RNA is a unifying feature for all Earth. ATP itself is a unifying feature for all Earth, for all life on Earth. So RNA world hypothesis has the potential to solve the age-old chicken or egg question. RNA can both store biological information and it can carry out enzymatic reactions. And it can also evolve. So we have one more last question to answer. Where did life begin? This question can be reduced to whether it happened on Earth or beyond it. The theory that suggests that life could have been delivered to our planet is called panspermia. There are actually several variants of panspermia. However, the panspermia theory does not answer the question of life's origin, it just puts it aside. Therefore, for the purpose of our discussion, let's just assume that life did start here on Earth. So the universe is full of organic matter. It's estimated that about a ton of carbon-made molecules fall on Earth every year. This should come as no surprise when we realize that carbon is the fourth most abundant element in the universe. So the building blocks of life can be synthesized from simple gases like Miller-Urey experiment, or they can be delivered to Earth. And now they have to assemble into a bigger structure to form living cell. But where did life first appear on Earth? Where did this assembly happen? Probably somewhere where a free source of energy was available. And the source of energy could be our sun. It could be geothermal, for example, volcanoes or even underwater volcanoes. And maybe again, the clues needed to answer this question can be found in our own cells. We know that all living cells use ATP to store energy. So let's see how the ATP is produced. To produce ATP, cell accumulates hydrogen on one side of a membrane and then enable flow in only one direction, and using this directional flow to move what looks like a water mill. This process creates a high energy bound in ATP. This mill that you see here is a protein, an enzyme called ATP synthase. But the first cells couldn't use enzyme like ATPase, like ATP synthase, because it is a complex, highly evolved protein. But maybe the other component, the gradient of hydrogen, might actually be older than the enzyme itself. On Earth, hydrothermal vents are free source of hydrogen. These underwater chimneys are covered with microscopic pockets that could serve as molds and hold the first cells through which the hydrogen would be passing for free. We also know that fat molecules in water, they can easily form capsules that look just like cells. The truth is that we don't know where life originated. And there is a lot of speculations about where. There is even a theory about condensate. The atmosphere was reduced enough that the origin of life could have happened in the whole atmosphere of Earth, if concentrated enough. But for the purpose of our discussion, let's imagine that the first life was formed on the surface of a hydrothermal vent. So the geothermal energy released from the vent makes it possible for RNA-based enzymes, ribozymes, to evolve. But the path to life, as we know, still remains very long. The primitive life has to replace information stored in RNA with DNA. Instead of using ribozymes, it has to start using proteins. It has to develop efficient ways of storing energy. Eventually, the last universal common ancestor evolved, and we are all genetically related to it. There is some clues about what life could have looked like before Luca, and these secrets are hidden in the genetic code itself, how this code emerged. But our journey ends here. The story I just told you has a lot of missing parts, but it's based on solid foundations. The foundations are made on the processes that we either discovered or we can recreate in our labs. And in conclusion, it seems that life is something that just happens and will continue happening as long as right conditions exist on Earth or anywhere else. So understanding of the process of the origin of life, of distribution of life, and the future of life in the universe is the main subject of astrobiology. And as I mentioned, we recently created the Astrobiology Society in Poland. We started gathering people from very different parts of Earth. So we have, but mostly speaking in Polish. We just recently introduced one seminar a month in English. So we're now open for collaborations from outside of Poland too. And here are some subjects that, like working groups that we created within the society. So of course, there is the origin of life. There is history of life on Earth, how it evolved. We have planetology and astrophysics groups. We have organisms of extremes, so the extremophiles that show us the barriers of what life can sustain, what are the limits for living organisms. We have a group of cosmic medicine, of just medicine for space medicine. So the health of astronauts, for example, how it changes on the orbit, lack of gravity, lack of magnetic field seems to be important. Space architecture, we have quite a few architects on board. Synthetic biology, that I will be talking a little bit more in a moment. Analog missions for analog astronauts missions. Search for extraterrestrial intelligence, future missions. alternative biochemistry, and even cosmic, like law, the law in the aspect of astronomy and space exploration. So astrobiology is not only those subjects, but it's also a philosophy that offers this unique perspective that Earth is a spaceship, and we all sit on it at the helm of this spaceship. And in order to be able to lead it as long as possible, we need to cooperate on a global scale. And I think that astrobiology is a great vehicle to integrate people with very different specialties. It's an extremely interdisciplinary science, and it helps to bring us all together with this common goal to carry about little planet. So little, here are some emotional photos so you can see how small it is. Of course, very well known for a lot of astronomers. Image of Earth from the orbit, Moon, from Mars, and from the vicinity of Saturn. So I personally ended up in NASA Ames Research Center, where beginnings of astrobiology happened, I would say. Ames is probably the closest to a university out of all NASA facilities. There is a lot of basic research going on. This is where Carl Sagan was working. And I have pleasure to take part in experiments that eventually end up on the satellites or on ISS. But we also study life as applications, like how we can apply life for space science, something that is called a synthetic biology. So the use of biology as an instrument, as a tool. And it is possible because biology is pretty much information, and as it is an information, we can edit it. It is not so complicated. It's just a bunch of simple letters that any computer can easily store. And with enough computational power, we can start recognizing common patterns. And we use this to our advantage, just cutting out certain functions that are coded in DNA and using and putting them together in various ways, almost like Lego blocks, and making non-living systems almost alive, but just producing what we want, for example, proteins. And also, the whole life on Earth is just a wonderful repository. If you look from the perspective of synthetic biology, it's just a billions years old repository of inventions and solutions that are stored in a readable format. So you could think like that if I can build something, I don't understand it. And we certainly can't build a cell. But as Drew Andy noticed, we don't need to understand biological system to be able to design specific functions. And that's what we capitalize on. A very simple example and on time example is the PCR reaction. This is actually a biological solution that is enabled by polymerase, identification of polymerase. That is a protein. And we use it, for example, in diagnostic tests, using this enzyme so we can make billions of copies of a sequence of DNA within 1 hour. And of course, in a space exploration, everything boils down to the cost. And that's the main reason for synthetic biology. So bringing about 1 kilogram of payload from Earth to Mars cost about $3 million. So important strategy for NASA and many other agencies is to use in situ research utilization. So use whatever we can find there to build what we can so we don't have to bring it with us. And biology is extremely good at doing just so. So one of the awarded programs that was sponsored and is still sponsored, we are actually right now in the NIAC phase 2, so NASA Advanced Concepts. they're very, very basic research, usually starting with just a proof of concept, but we already proved the concept and we are on the developing. It's called microarchitecture because we use the ability of mycelium, so just fungi, to build structures. And we can feed these fungus with, for example, cyanobacteria. And cyanobacteria, with a little bit caring and a little bit tweaking, even genetic tweaking, is capable of growing in Martian conditions, isolated, of course, in a habitat, but it can assimilate carbon dioxide and build its biomass. Then the fungi can grow on it, and it's extremely resistant structure. It can take up shapes, and then we built several constructions made of the fungus, and it really worked well. So the concept is to build a base on the Mars, maybe using the organisms like fungus. It goes completely against the planetary protection, so I'm not a big supporter of this concept, but maybe in the future when humans are there, it's Mars already going to be dirty, humans are dirty microbiologically. And another concept and the project that was awarded is astropharmacy. Astropharmacy is using a non-living system that is made of just liposomes, where we take a liposome is just a lipid bubble, and we take components of cells that are needed for protein production. We get rid of most of the bacterium, leaving just single proteins and some DNA, and we mix it and we can lyophilize it so it can be dried out, and it's very storage safe. It can be stored for very long, much longer than the product that is a protein can be stored, the protein-based drug. So the idea is to use the components of cells to produce, for example, drugs, the protein-based drugs on demand. So one thing is that we don't have to deal with storage time, or called shelf time. So how long the reagent can sustain storage, but also it gives us a possibility that if we have a robust system for DNA synthesis and then a protein expression, for example, on Mars, we don't need to deliver drugs. The only thing that we have to send is the information how to make these drugs, and the information can travel with the speed of light and doesn't wait. So ultimately, there will be labs on chips used in space. So with this, I would like to finish when reminding you about our society. Please go and visit AstroBioPL. It's pretty easy to remember, AstroBioPL, and maybe you will find something that you like, and maybe you will make connections within the society, and we can work on projects together. One of our biggest initiatives right now are conference. We did one conference last year. We're going to make another. It's a Life in Space conference, and the other one is course in astrobiology. And part of the course is going to be astronomy. So we would appreciate your help with preparing the course. With this, thank you very much for your attention, and let me know if you have any questions.
Thank you very much, Tomasz. I'll do the clapping on behalf of everyone. Thank you very much. And so we are here on the Zoom only meeting. So if you have a question, please raise your Zoom hand first and then speak up. Milena, please.
Hi, Milena.
Hi, Tomek. Nice to see you. Thank you for a great talk. Could you just summarize, as we are mostly an astronomers group, if we go and talk about life in space, could you just summarize the recent, let's say, research on life on other planets, not only planets, celestial bodies in our solar system? Because there were stories with phosphine and not phosphine.
Oh yeah, okay, yeah, that's a good, very good question. I mean, yeah, that's a lecture for its own, but let's go. Let's make it quick and efficient. good that you mentioned phosphine on Venus. So first of all, the destination for looking for life was so far mostly Mars. And since the Viking probe was unable to detect life, it was the outcome of the experiment was inconclusive. We couldn't really make anything out of it. NASA shifted into follow the water. So the missions that followed the Vikings were looking for water on Mars. If in history there were conditions for life. And now with the Curiosity, we know that the water was abundant and was present. There was a very good time of period on Mars when life could evolve. So now the Curiosity, the Perseverance, the next one, is actually going on Mars and collecting samples on a slope of a crater, going through different geological times of Mars, collecting samples and leaving them behind. Because the biggest endeavor of NASA so far is the Mars Return Sample Mission. And the Perseverance is the first stage of it. It's going to be a mix of ESA and NASA missions that are going to have to land again on the Mars, collect the samples, shoot them on the orbit. There's going to be ESA-made satellite that is going to capture the sample. That is an ingenious, incredible thing, because they want to make a second and a third layer of containment on the samples without touching it in space. So very precise navigation there. And they want to have this covered wrap it up so many times, because they don't want it to break open when they drop it on Earth. And that's the plan. They're going to drop it on the desert and hope it's not going to break up. And that's when we actually going to learn the most, because being able to use facilities on Earth is totally different than trying to make experiments on Mars itself. So Mars, that's the story about looking for a search for life on Mars. Of course, there are other missions for the moons, like the water worlds. There is Europa Keeper that is coming up. Cassini detected incredibly big carbon compounds, much bigger than its spectroscopes were designed to detect, to analyze. So we can't say what it was. We just know that there is an incredibly complex organic matter there. So it might be something. We know that there is a hydrogen, particles of hydrogen in the plumes. So that means that their water has interaction with mineral surfaces, and that's important for life. So there's an energy exchange, and the most fundamental organisms in ecosystem can use this kind of energy for a long time, and then maybe a bigger ecosystem is built on that. So there is still hope for life on the water moons. Titan is an interesting destination too. There is some research on Earth made, for example, by Stephen Banner, who works on alternative biochemistry. So we're thinking about different solvents. In this case, on Titan, we have carbohydrates instead of water, and maybe the life could evolve in this crazy environment. It would be nothing like what we know today here, but Stephen Banner was able to show some different folds of DNA and even different composition of of DNA, that he could build coding strands of DNA in these conditions. So theoretically, it's possible. And there is this Dragonfly mission that is going to be a big drone. Titan's atmosphere is really dense, so there you can build a very heavy flying machines there. So that is something exciting for astrobiologists too. So let's go back closer to Venus. So phosphine is actually one of the discoveries of our member of our society, Janusz Pętkowski. I'm sure he's going to give a talk pretty soon, because they just published another paper. Long story short, they first found phosphine. People speculated that maybe they had problems with detection and so on, but every of these arguments seems to be so far withstand. They withstand those arguments that maybe there were some mistakes. They actually reached out to very old detections from Venera and the times when the Venera was sent to Venus. They had to translate from Russian to be able to read this data. And what they did, they found out that actually phosphine and other signatures for their model were already detected during those early missions. They just kind of ignored it because they didn't fit in the currently used atmospheric model of Venus. So Janusz and his team, and Sarah Seeger is the first author and the lead of the team, they created a new model for atmosphere of Venus based on those new findings and old findings all together. And right now, NASA approved 2 missions to Venus, an orbiter, and I think there is a lander too. And also the very first time in the history, a private mission to another planet is planned. MIT, with collaboration with Breakthrough Initiative, they are building, and they just released a concept mission on a paper. It's a 100-page document, a Venus Life Finder mission, where they would be collecting samples and looking for life not necessarily similar to what we have. The premise is that in Venus, there is, of course, this awful atmosphere of sulfur, but about 1 kilometer above the surface, the temperatures are mild, similar to temperatures on Earth. And the new model shows that there might be pockets of water distributed within the clouds. So there could be like water vapor groupings where life potentially could evolve. But it would be the first kind of life we know that is not attached to Earth. So on Earth, we know microorganisms live in clouds, but they divide on Earth. No one showed division of cell in the cloud. So we don't know how it would work on Venus. And of course, the last thing is a Space Webb telescope that is going to look for biosignatures in atmospheres of exoplanets. And that's another exciting thing for astrobiologists.
Thanks a lot for a very comprehensive answer. So yeah, that's great.
Thanks.
Thank you, Milena, for this question and the answer of Tomek. Any other questions from anyone else? Pavel.
Then maybe me. Hi again.
Hi.
Yeah, very nice talk. I have a question about the ribosome and the self-replication.
Mm-hmm.
And you said that this is very important in the context of natural selection. And is it the same as the genetic modification, or is it somehow related with that?
Well, mutations are usually random. What we do in the lab, we can actually be very, very precise. You probably heard about the CRISPR method. We don't use it for RNA, but CRISPR is one of the examples of incredibly precise methods to modify DNA, even in a very complex cell as eukaryotic cell that has a lot of repair mechanisms that would prevent it. But CRISPR can do that. So RNA, one thing that I'm going to add to self-replication of RNA, the one single strand is not enough to copy itself. It needs another one. So they both have to work on each other to copy each other. And that's the little detail that I think it's important. And the mutation, well, just the replication is not ideal. Even in our cells, highly evolved polymerase that has 3.7 billion years or so, it still makes every million or about 100,000 or a million nucleotides, it actually puts a wrong one. But then there are at least 3 or 4 mechanisms how to repair that. So it is a constant balance of mutations and then repair mechanism. So in the RNA world where there was no repair mechanism, the variation must have been incredible, incredible variation because of mutation. And also, even if we imagine a primitive cell made of just lipid bubble, there were no mechanism that would prevent from foreign DNA to get inside or your DNA of leaving the cell. So the exchange between the protocells would look more like an evasion and not and not just exchange, right? So like right now, we are very protected to what is our content of DNA information, our cells protected. But at the beginning, before Luca, it must have been total mess. We couldn't even say it's an organism because it didn't have its own DNA. So, mixed together. Does that answer your question?
Thank you very much.
Thank you. I don't see any more questions. I just have a brief one. So how about Antarctica? Is it of interest for astrobiologists?
Absolutely. I mean, first thing that I think about Antarctica is that underneath the ice, there is so much land with so much biodiversity that is just hidden from us, right? So Sławomir Tułaczyk, he's one of the members of the Astrobiology Society. He's a glaciologist. He actually did several studies on Antarctica, and they drill into like ancient lakes looking for life. And they did find cells, but they still think these are, they came through underground from the ocean and somehow got to the lakes. And interesting is how they drill in ice. They use water. So the biggest and the heaviest thing they have to bring with them are filters. So they know that the water they're passing have absolutely nothing in it, right? Yeah, so Antarctica, definitely an interesting part. Another thing that comes to my mind, aerobiology. So we talked about life in clouds. Antarctica, there are the winds. If you send a balloon, the winds keep the balloon over more or less the same spot for a long time, and they can be on a high elevation, so it's a good analog for high radiation and stuff.
Cool, very nice. Thank you very much, Tomasz. That was really enlightening and very inspiring, I believe, for many of us. So thank you very much.
Pleasure.
And thank you all for joining. And that was, as I said, that was the last seminar this this semester. And we begin again in March. So have a nice break and bye-bye.
Thanks a lot.
Goodbye.